recording before I do attendance so we can get it all documented. Excuse me. Van Cross, Taylor, Michaela's here. If I call your name and you're wearing WVSU gear, make sure you let me know. Reed, Redmond, Hemmings. Gotcha. Of course, you're all getting a little bit of extra credit for being here physically. As of the week prior to the spring break, that's the new thing. Anyway, Bowers, Asbury, Tudors here in WVSU gear. Okay. Felder, Hudson, Burdette. Finally. Newton, Massey, Richardson, Cedric. Bryce is here in WVSU gear. Pauly, Midkiff is here, also wearing WVSU stuff. Eric, Sipple, basically everybody. We have Richardson online. Good morning. So Hudson is here. Taylor's online. Got a little less than 3 minutes. Do some, I guess, less important announcements / reminders. So, as of Sunday, that's another week. So you should have about 57, 58 points of independent work by now. So make sure you get to that, especially if you haven't written any papers yet. I think there's a few people who have written 0 papers. You're definitely going to want to at least write 1 paper so you know how I grade. Because for those of you who have written a paper, you realize it's probably not as straightforward as you might think, right? Like me turning everything in one big paragraph and maybe me deleting these things and not counting those things. So please go ahead and send the paper if you haven't done one yet. What else for easy reminders? Hold on, let me get Massey here. You wearing WVSU stuff? Yep. Oh, no? Yeah, okay, sorry. I'm not wearing my glasses. And it's dark. So anyway, yeah. Independent work. Definitely want to work on getting it done. Speaking of which, that's a good segue to remind you can get points for doing the study guides, those study guides that I use to write the exams. So since we just finished Chapter 8, that might be a good time to do the study guide for Chapter 8 if you haven't done it yet. Likewise, it's a good idea to go ahead and start looking at the study guide for Chapter 9, which is what we're about to jump into. That way, hopefully those questions will be in the back of your mind. And as I'm going through a lecture, maybe you'll get those answers that you're looking for. And or if I say something during the lecture that, confuses you based on those questions, you can pause and say, hey, can you explain this for me? See, Cedric is online. Good morning. So yeah, independent work, study guides. And also, again, like I said, we just finished Chapter 8. Today we're going to start Chapter 9. After this will be Chapter 10. And then after that will be the 3rd exam. So we're moving right along. And then after that, obviously, will be the final exam. So, 2 more chapters for this exam, and then 3 more chapters for the final. What else in our less than a minute that we have here? Lab, if you haven't done the pre-labs yet, please go ahead and do them. I'm going to deduct fewer points if you get it done before you get to lab, because it's important to do pre-labs, right? Because it's supposed to prepare you for the lab. And considering this is a big team effort this week, it's even more important for you to be ready for lab. Otherwise, people are going to be waiting on you if you're like the weak link or the slowest person, right? So make sure you do the pre-labs. What else? I guess that's it. Does anybody have any questions? Any ideas for independent work or extra credit, whatever? All right, then I guess my last announcement / reminder, since it's still not 8, make sure you guys pay close attention to details. So read my announcements carefully. take a look at the spreadsheet occasionally. Read the syllabus if you haven't yet. I know it's pretty late in the game, but I have reason to believe most of you have not read the syllabus still. Don't ask me how I know, but I know. Anyway, it is now 8 o'clock. Does anybody have any questions? All right, I am going to wait until 8:01 before we officially get started. Oh, yeah, I guess attendance. Obviously, I haven't posted the last two videos. It's just the first 8-week semester finished last week. So I've been really busy wrapping that up because obviously we are doing a 16-week semester, but I have 2 online courses that just ended. So that had to take priority. So hopefully I'll get those videos up for you. And hopefully today's records as well, and then I can get this one up as well. So no questions? All right. So this next chapter is a really good segue into what we just finished. When we talked about cellular reproduction, right? Mitosis and meiosis. We talked about what happened with the homologous chromosomes and the sister chromatids. And that all that, and we talked about genetic variation. All that information is actually going to be relevant to this chapter. So the title of this chapter is, as you can see, Patterns of Inheritance. But essentially, you can just call it genetics. That's what you're going to be learning about is genetics. Pretty light. You're all right back there, Asbury? All right. Anyway, I guess the first word for attendance, since I'm talking about it anyway, is bright. Like the word bright, like the bright light that's shining on her back there. So don't do anything sneaky. All right. Why genetics matter? Let's do the little guessing game that we sometimes do. So how would you describe her hair color? I would too. Good. My wife would probably correct me. There's probably some other more specific name. Like, who's mauve or something? I don't know. Anyway, yes, I would call that red. But do you think she was born with that hair color? Okay. I would agree too. We don't know for sure. What's right here on her arm? That's my assumption too. Either that or a really crazy birthmark, right? But yes, probably a tattoo. Looks like she has a whole sleeve of tattoos right there. All right, so obviously she has red hair. She's got some marks on her skin. Do you think if she were to have a child, a baby, the baby would be born with that color hair or with the same markings on her arms? No, I agree. Probably not, right? Why? She has it on her. It's because that stuff's not heritable, right? So what you're going to learn, one of the major things you're going to learn in this chapter, and then it'll be important for future chapters, is that for genetics, it has to be heritable, right? So yes, you can make changes to your body or to your genome even sometimes. We'll dive into that later. It has to be heritable for it to be passed down from generation to generation. And when we're talking about genetics, that's what we're talking about. Mostly that's what we focus on, at least in this chapter, is again, like the inheritance. So yes, we could talk about genetics, and we do a little bit, like when looking at your genetics and looking at how your genes are functioning and how they're producing proteins, and we will talk about that a little bit. And then we're also going to talk more importantly, in the context of this chapter, of how those genes are passed along. Does that make sense? So it's one thing to look at your genes and what they're doing, and then it's another thing to look at how you are passing along genes and how you're combining genes with other people. So we'll talk about both of those things in this chapter. Speaking of which, this is a little baby girl. I know it's kind of hard to tell, but any idea why there's a picture, what this picture, what they might be getting at with this picture? It's a real shot in the dark. I just want to see if anybody can guess. That's a good guess. I'll just go ahead and tell you, though. It's a picture of a father and his daughter, because it's the father that determines the sex of the child. Because as you know from last chapter, a male has an X and a Y chromosome, and a female has 2 X chromosomes, right? So no matter what, the female is only giving an X chromosome. The father, the male, could either give an X or a Y, and that determines what the sex of the child will be. So that's usually a little bit counterintuitive. So keep that in mind for the exam. Speaking of which, if you've done the pre-lab video, and if you haven't, please do it. Not just for the lab, but also to help prepare you for this chapter. But they talk about the gene for male baldness. That actually comes from your mom, which is also a little bit counterintuitive, right? But not that that's important for the grand scheme of genetics, but small little tidbit there. I don't actually remember what your picture or what your book was getting at with this picture. I think basically they just talk about, if we're talking about genetics and heredity, talking about how you combine genes and features, and hopefully you look at that picture and see that there's some similarities between those folks and also some differences And you guys probably already know all about that when it comes to heredity, right? You're not a perfect clone of your mom. You're not a perfect clone of your dad. You're a mix. Now, granted, you might look more like your mom or your dad, but you're still a mix of the two. If I remember correctly, that's what your book was getting at for that picture. If I'm wrong and you read it, let me know. I'm not going to go back and read it. It's not that important for the grand scheme of this course. So any questions about this slide? Now that we're done with the little introduction, let's jump into this, biology and society. Again, as always, every chapter starts with this, and I always skip it. If you're into dogs, I would highly recommend reading it because it is slightly interesting. Actually, I would read it anyway because this also has implications for next chapter. Excuse me, not next chapter, but the chapter after that when we start talking about evolution. But yeah, this is just, to me, slightly interesting. I like dogs. I don't have any, but I do find them interesting. And that video I just posted right before spring break, I think it was. Again, if you're into dogs, you should watch that video. It's not going to teach you anything specific that's going to help you on any exams, but there are some biological terms and concepts that will help reinforce what you've learned here. And again, it's about dogs. All right, here we go. Let's jump into the meat and potatoes of this chapter. We have 3 main bullet points here. As you can see, it's a long one. The first one is about genetics and heredity, the overview. So as you can see, again, we've got a lot to talk about. Then finally, we'll get into some variations of all this stuff I'm going to teach you. So I'm going to teach you all this stuff, and then I'm going to get here and say, well, hold on, it's not quite that simple. And then finally, in a sense, I'm going to repeat myself and talk about everything I've said, but from the context of chromosomes. But actually what I'm going to try to do, knowing that this is coming, is as I'm teaching you guys all this, I'm going to use the chromosomes as examples. So hopefully by the time we get to that 3rd bullet point, we can just fly right through it because it'll be no new information. So here we go. Genetics and heredity. First of all, you need to know what heredity is. Heredity is the transmission of traits from one generation to the next. Pretty simple. You need to know that for the exam. Obviously, as usual, I don't usually ask you these things verbatim. So understand the definition, not just the words and the order that they're in, if that makes any sense. That always used to irritate me with vocabulary classes in high school. Like you'd have to memorize those definitions word for word, but you could do that and not actually understand the definition. Anyway, that's heredity. Any questions about what heredity is? All right, the next term you need to understand is genetics, which is the scientific study of heredity. Pretty simple. And I'm actually probably, I might ask you the definition of heredity. Let me write on here. I might ask you that. I almost definitely won't ask you the definition of genetics because there's just going to be so much about genetics that you need to understand that I'm just going to assume you understand the definition. So I like the way your book teaches about genetics. So the guy basically figured out modern genetics as we know it. His name is Gregor Mendel. And your book kind of tells the story of Gregor Mendel and how he figured out what he figured out, which is good, I think, because basically that kind of brings you back to his level, right? So you start off knowing nothing about genetics, and then you're going to learn things as he learned them. However, that being said, I'm not going to necessarily give you a history exam, so I'm not going to be asking you his name or when he argued what he argued. So I'll let you know the important information as we move forward, but just know that for the most part, I'm not going to ask you questions about him specifically, because this is not a history class. Anyway, back in the 1860s, he argued that parents pass down their unique genes to the offspring. There's no need to write that down. This is just an introduction. That's what he argued. We're going to learn what his argument was, right? And that's the kind of stuff you're going to need to know is what his argument was, what he used to support that argument. I put a little star next to the word genes because back then they didn't have the word genes. And of course, this was brand new. So back then, I think he called them heritable traits. Not that that matters either. But yeah, it is the genes that are responsible for the inherited traits. And genes, he also argued that genes retain their individual identities generation after generation, no matter how they're mixed up. And again, nothing to write down yet. This is still an introduction to what you're about to learn. But back in the day, we all know this. Probably everything I'm telling you right now, you're like, yeah, I knew that. But back in the day, they didn't know that. Yes, they knew things were inherited, but he didn't know there was actually individual genes. And again, he didn't call them genes, but he knew, he figured out that there was something inside cells that was responsible for all this. He also argued that genes retain their individual identities generation after generation, no matter how they're mixed up or the mask. Already talked about this, but basically, yeah, we'll get into that. So again, this is all just basically a big introduction to what we're going to talk about. So, any questions about this introduction? All right, let's move forward. He did his work in a place called Abbey Garden. Not a history class. I'm not going to ask you that. But again, I like the way your book presents this information. It tells it as a story. It's a little bit more interesting, I think, when you get it as a story. Mendel used garden peas in his studies. Not that I'm going to ask you that, so I'm going to quit. Therefore, why he did it's not that important, but we'll talk about it quickly because you booked it too. Peas are easy to grow, and they come in many readily distinguishable varieties. Right, so you might not want to grow. Has anybody ever tried to grow orchids? They're just like really temperamental plants. They're easy to kill. Or you wouldn't want to grow a redwood, right? Because first of all, it's going to take it hundreds, thousands of years to grow up. And then, like, where would you put it, right? Not to mention the fact that if you're talking about genetics, you need to be having multiple plants and multiple generations. So, yeah, that's why he used peas. Also, another good example about the whole readily distinguishable varieties. Let's say, for example, I don't know, you wouldn't want to use crabgrass. Even I look at the grass and look at that looks just like that one, which is just like that one. Like, there's not readily distinguishable varieties. Anyway, here's some terms you need to know. Character versus trait. You need to know their definitions, the relationship between those terms, and how they're different. Not necessarily because I'm going to ask you on the exam, but because I'm going to use these terms a lot in this chapter. And you need to know what I'm saying when I use them. So character. That is a heritable feature that varies among individuals. So hair color is a character. Eye color is a character. Height is a character. Certain diseases, in a sense, are a character. Like, do you have the disease or don't you? Any questions about what a character is? All right, the next term is a trait, which is a variant of the character. So again, hair color is a character. Brown would be a trait. Red would be a trait. Blonde would be a trait. Eye color would be a character. Blue would be a trait. Green would be a trait. Brown would be a trait. So yeah, make sure you understand those terms. Just that way when I use them, you understand what I'm saying. One thing that's important, not for the exam, but for his studies, each character that he studied occurred in only two distinct traits. So again, eye color wouldn't be a good one. Well, of course, he used peas, not humans anyway, but eye color wouldn't be good because, again, blue, green, brown, and then, of course, there's all kinds of variations of all those. Hair, right, even there, brown, red, blonde, and then all kinds of variations of those, right? So those don't come in two distinct traits. But the stuff he did come in two distinct traits, which made things much more simple for him. And for figuring things out on the exam, it'll also make things a little bit more simple for you. Anyway, any questions about this slide? All right. An important advantage of pea plants is that he could strictly control their reproduction. So again, we're just telling the story of what he did and why he did it. So there'll be no questions about what I'm about to tell you here. And again, I teach out of the book. So here we go. The petals enclose the egg-producing organs, which are called the carpels, and the sperm-producing organs, which are called the stamens. Another way of putting this first bullet point is that if you look at a flower and it's all closed up, at least for pea plants, all the parts are there, right? The male parts, the female parts. So if you look at a pea plant and look at the flower, it is both sexes, right? Both sexes in that one flower. So consequently, pea plants usually self-fertilize, right? So they don't usually need to share pollen with other plants or even other flowers on the same plant because it self-fertilizes. A weird way of thinking about this, and this is not necessarily correct, but it's probably a weird vision that'll probably never come out of your head. Imagine if you had both male and female parts, and when you're wearing your underwear, that's the same as everything being covered up with the petals. And if you're not careful, you might get yourself pregnant. Same thing here, right? At least to an extent. Any questions about this slide? All right, let's do underwear for the next independent work. Attendance word. Underwear, since we just said it. So here's a picture of everything I'm saying. There's the pea flower, or pea plant flower, right? You've got the female part, the male parts, You got the petal. And this is kind of opened it up, right? So when they're in bloom, everything's open, but when they're closed, it's all right there next to each other. So again, you can have that self-fertilization. Also, yeah, when you think about it, for independent, I think I asked this on the biology group on Facebook for extra credit or independent work. But basically, pollen is a bunch of sperm. It's plant sperm everywhere because we don't get much of it here in West Virginia, but in places like South Carolina right now, they're getting a lot of pollen. Keep that in mind. That's just plant sperm everywhere. Or when you're smelling like, smells like flowers out here. You're smelling plant genitalia. Fun stuff. Nothing to do with genetics, but let's keep moving. All right, to fertilize one plant with the pollen from another plant, he did it by hand, right? This way, he was always sure that this plant was crossing with this plant. He didn't want to leave anything up to chance, which makes sense because he's studying genetics. So here's how he did it. You can see he's cutting off the male parts there, using a little paintbrush to move the flower from move the pollen from one flower to the other. Then that turns into the fruit, i.e. the peas. So we have all those peas, and then he can take those peas and plant them and then get new bushes. So there you go. That's how he did everything. One thing that will be important that I'm going to bring up later, but you don't really need to know it yet, is the difference between the P generation and the F1 generation, but we'll come into that later. Other than that, there's nothing important as far as the exam is concerned on this slide. Even if you don't know how he did this or why he did it, that's okay. You're still going to need to understand and still will be able to understand the results of his studies. All right, here's another term you need to know, and I think this is like one of the first questions on the study guide for Chapter 9. He created what's called purebred varieties of these plants. What is a purebred? A purebred is something that self-fertilizes to produce offspring identical to the parent. So yeah, he did all that cross-pollination that I talked about where he controlled it, but then he did it long enough to where he could get a plant and have it self-fertilize again, and the offspring would be identical. And this will make a little bit more sense later. But like I said, everything he did had two different traits, right? All the characters had two different traits. So, for example, purple flower versus white flower. So before he even started his study, he did like generation after generation after generation of producing these purebred varieties. Or if you've already looked at the lab, you could almost just call those the homozygous, right? Those purebred were all homozygous dominant or homozygous recessive. Of course, he didn't know those words yet, but that's what he did. That's what a purebred is. For whatever gene you're looking at, and again, these are terms he wasn't using, for whatever gene you're looking at, those purebreds are homozygous, whether it's homozygous dominant or homozygous recessive. They're pure, right? They only have that one allele. Anyway, you need to know what a purebred is. So to do his study, once he finally got a bunch of purebred plants, then he could get to work, right? So he crossed two different purebred varieties, and that's what a hybrid is, as the offspring of two different purebred varieties. So again, sticking with the same example, he could have a purebred purple flowered plant crossing it with a purebred white flowered plant, and that would be called a hybrid. Right? The hybrid is the noun, right? That's what he created. The verb of doing that thing is called a genetic cross. You'll need to know that term because I'm going to use it in this this chapter quite a bit. So I might not ask you what a genetic cross is, but again, you need to understand the term because I'm using it. The plants that he did do that initial genetic cross with, those initial parents, those purebred parents, are called the P generation. And to help you remember that, you can think of it as the parental plants that starts with P, or the purebred, right, that starts with P. Either one, whatever helps you remember. That's the P generation. So the P generation is crossed, those two purebred plants, and then they have that first generation after that, and that's called the F1 generation. And then he could take the ones from the F1 generation and cross them with themselves, and that would bring the F2 generation. And that's as simple, or that's the extent of what your book talks about. So just know P, F1, and F2. Of course, you have to imagine there's so many different ways you can work with this. You can do like a P generation, and then take the F1 generation and cross it back with the P generation, or Same thing here. Once you get down to the F2 generation, you can cross that back with the F1 generation or cross it back with the P generation, but we're not getting into any of that. We'll just keep it nice and simple. So again, you cross the P generation. The result of that is the F1 generation, and you cross those with each other. The result of that is the F2 generation. And again, these are terms I'm not necessarily going to ask on the exam, but I'm going to use them a lot, so you need to understand what they mean. Any questions? All right, let's talk about Mendel's law of segregation. So in the last chapter, we talked about something called independent assortment. And I said this is going to be kind of simple at first, and it is kind of simple. The concept of independent assortment, I think, is kind of simple. But now I'm teaching you something completely different called law of segregation. And as a student, as a young student, not wasn't young, but as an early student, those two things confuse me. Is this because they sound close? Right? Independent assortment and segregation, like are we assorting them or separating them, right? They sound so close. So as I'm teaching these things, keep in mind that you do need to know the difference, and keep in mind that they are a little bit complicated sometimes just because the names are so close. But let's talk about the first one, Mendel's law of segregation. Again, he performed experiments, and in these experiments, he tracked the inheritance of characters. For example, flower color, right? So again, purple or white flowers. This is an example. And again, like I just said, they occur in two different traits. So purple or white is the example I'm giving there. And these results, the results of these experiments he did led him to formulate several hypotheses about inheritance. So let's talk about these hypotheses. Well, first of all, here's a picture of what we're talking about. Not that you need to know. You definitely don't need to study this. But again, I keep using this example. The flower color character comes in two traits, purple or white. The flower position comes in two traits. It could be at the very tip of the plant, which is called terminal, or it could be somewhere else, which is called axial. Seed color comes in two traits, yellow or green. Even the smoothness, right? It could be round or it could be wrinkled. All these things. We're really only going to talk about the flower color, seed shape. Not that it matters, but for some reason, genetics books, that's usually the examples they use. But he looked at all these things. All right, here we go. Here's another term you need to understand, and we're going to come back to his hypotheses shortly. But a monohybrid cross is a cross between the purebred plants that differ in only one character. So I already told you earlier what the P generation is, right? I said there's a purebred, they're hybrid, or they're purebreds. So they're basically homozygous, recessive or dominant, whatever. They only have that one allele. And I told you when you cross those things, that's called a genetic cross. Now I'm getting even more specific. So under the category of genetic cross, which is when you cross two purebreds, if you're only considering just the one character, then that genetic cross is called a monohybrid cross. So just remember, mono means one, right? Because the only other option, the only other thing I'm going to teach you about later, it's called a dihybrid cross. For the multiple choice exam, I'm not going to try to trick you up and have like monohybrid or monoplant, right? Nothing like that. It's going to be monohybrid cross or dihybrid cross. So if you can remember mono equals 1 and di equals 2, you should be good there. Anyway, the example we keep using, flower color, right? So if you have a purebred purple flower, cross it with a purebred white flower plant, that would be a monohybrid cross because you're looking at just that one character. And when he did this, using the color as an example, he found out that next generation, right, the F1 generation, it had all purple flowers, which had to have been, I don't know, not confusing, but interesting at the very least back then because they didn't know anything about genetics. So think about that. You had all these white flowers, purple flowers, plants. You cross them, and no matter how many times he did that, they always had purple flowers, always. He never had a single plant with white flowers. So again, that was his action. That was his observation. That led to a question. And the question was the factor for white flower lost as a result of the cross? In other words, where did the genes for the white flower go? Of course, again, he didn't call them genes, but that's okay. And that's a logical question, right? If he did this hundreds of times or even thousands of times and never, ever, ever, ever got a white flower, then right, you got to wonder what happened. What happened to that white flower gene or allele? But then, so again, crossed the purebred parents thousands of times. Every time he had thousands of the next generation, which is the F1 generation. They all had purple flowers every time. And then he started crossing those F1 generations to bring up the F2 generation. So then he found out the answer to his question was no. The gene, again, or the allele for the white flower was not lost. And we'll talk about that moving forward. But for now, we can just know that the F1 was completely purple. And he was wondering, was the gene, was the allele, was the factor, was it lost for white flowers? So any questions about this slide before we further the conversation? Here's a visualization of what I'm saying. Of course, your book really simplifies it. It's not like he had one purple flower plant and one white flower plant and then crossed it and then had one purple flower plant. No, right? He did this thousands of times. So this is, again, this is very inaccurate when it comes to numbers. The results are accurate, but the numbers are very simplified, right? So this happened thousands of times. So anyway, the entire F1 generation, no matter how many times he did it, always a purple flower. Always. Never had a white. So again, he's thinking, well, what in the world? What happened to the white flower? Or what happened to the white allele? Or the white factor? So then he crossed these. He took an F1 plant, crossed it with another F1 plant. Or in other words, he took a purple plant, crossed it with another purple plant. Whoops. And then he found this ratio. And this is a ratio you need to know. About a 3-to-1 ratio. And I say about because imagine, again, he didn't take one purple flower and cross it and then have like 4 plants, right? It was not that simple. There's a whole lot. He did this thousands of times. So let's just assume he had 1,000 F2 plants. What this means is there was about 75% of them were purple and about 25% of them were white if there was 1,000 plants. So what, about 750 purple plants and about 250 white plants. Not that you need to know those numbers. The ratio that you need to know, I'm going to come back to it. So for now, I'm just going to say you need to know the ratio. It's 3-to-1. If we're talking about this F2 generation, right, if we're talking about crossing these, and I haven't told you these terms yet, but these, again, well, I've told you these are homozygous. What I didn't say is that these are heterozygous, meaning they have one of each allele, and we're going to talk about this will come up later. But then 3 out of 1 of the that next generation had the dominant trait. And again, I haven't even said anything about dominant yet, but I'll officially introduce you to those terms here shortly. So any questions about this slide? And to you guys, this must be pretty boring because it's, early in the morning on a Wednesday and you're sitting here looking at it on a PowerPoint slide. But think about that had to have been interesting back then, right? Thousands of plants. He kept doing this over and over and always got purple plants. It's like, what in the world? Why, what happened to the white flower? And then all of a sudden, once you start crossing these, no matter how many times he did it, he got about a 3-to-1 ratio. That had to have been interesting, right? Because they're all purple. So again, they didn't know about genetics. So we know that gene was just, that allele was just hidden, but he didn't. So that had to have been pretty cool. Anyway, again, the gene for the white flower didn't disappear. And I'm teaching out of your book. I think a better term for this would be allele. I can't type, or I can't draw there. Let's just call it, there we go, allele. The allele for the white flower didn't disappear, right? It was just hidden when the purple allele was present. So that means we knew that the F1 plants carried 2, and your book keeps saying genes because that's just an easy way to think about it. But remember, it's an allele. And I guess I haven't officially told you what an allele is yet, but if you've done the pre-lab, you know what an allele is. So again, we had the parents that we crossed to produce the F1, right? So we know now, because of what he did, that this second generation here, this F1 generation, must have had 2 alleles, right? It must have had one for the purple. Obviously, it had one for the purple because 100% of them were purple. So we knew that they had the allele for purple. But then they also must have had an allele for white. Otherwise, the F2 generation, the one that came after this, couldn't have a white flower. Like, where did that otherwise, it just wouldn't happen. Any questions about this slide? All right, from these results and the results of some other things I haven't told you about, he came up with 4 hypotheses. The first one is there are alternative versions of genes that account for variations in inherited characters. So, again, basically, not using the same words, he figured out, like for the flower color, that was just one gene with different versions, right? There's one gene responsible for the flower color. There's 2 different versions of it. One of them said purple, one of them said white. Right, so that's the first thing you figured out. Any questions about number 1? Okay. Number 2, whoops, sorry. Here we go. Now I'm officially giving you this term, but again, if you've done the pre-lab, you should already be familiar with it. We call the alternative versions of these genes alleles. So there's whatever gene, and I don't remember the name of it, but there is just one gene responsible for the flower color of these pea plants, and it has a name now. But that one gene has 2 different versions. To throw back to what we talked about in the last chapter about what a homologous chromosome is, and I told you, right, they have the same genes at the same location of the chromosome. This is what I'm talking about, right? So for humans, if there's a gene for stinky breath on the 5th chromosome towards the middle, that's exactly where it would be for all of us. For your 5th chromosome towards the middle, for yours, and I'm making this up, but if that were the case, right there on the 5th chromosome, right towards the middle, there would be a gene for stinky breath. And there might be 2 different alleles, right? You might have the stinky breath gene or you might have the good breath gene, or excuse me, allele, right? You might have different versions. But either way, it's going to be located at the same location on the same chromosome. That's what an allele is. It's very important that you understand what an allele is. One, I might straight up ask you that term on the exam, but also so much of the stuff that we talk about, not just in this chapter, but future chapters. Alleles are very important for understanding what we're going to talk about. Next hypothesis. For each inherited character, so flower color, for example, an organism inherits 2 alleles, one from each parent. So even that F1 generation, because remember we went P, which is purebred, and then we had the F1. Even that F1 generation that was completely purple, they still had 2 alleles, right? Because remember, the parents of that F1 generation were either purple plants and white plants. That's right, that was the cross. So they had that purple allele, obviously, because that's what color they were. But they also got that white allele. It didn't show up in the F1 generation, but it was there. So any questions about number 2? Okay. An organism that has 2 identical alleles, like I've already said, but I'm officially giving you this word, is called homozygous. They have 2 of the same, right? So the prefix homo, you should think of same. Likewise, an organism that has 2 different alleles is called heterozygous. For the exam, you could just focus on the prefixes. I'm not going to try to trick you with like, is it a homozygous or homogeneous or homoallelous? I don't know. I'm not going to make anything up. You just need to know homozygous, 2 of the same alleles. Heterozygous, 2 different alleles. Obviously, and I've already hinted at this, the F1 generation all had to be heterozygous, right? Because they had 2 purebred varieties, a white purebred and a purple purebred. So obviously, each one was passing down 1 allele, so it had to have 1 white allele and 1 purple allele. Anyway, any questions so far? Oh, yeah. I didn't come up with this. I just found it. Had to share it. Heterozygotes. Just a little uneven. All right, number 3. I didn't come up with that either. I just find these things. I don't even go looking for them. If 2 alleles differ, right, so we're talking about being heterozygous, one of those alleles is going to determine the appearance, and the other one will not. So the one that does determine the appearance is called dominant, and the other one is called recessive. I'm definitely going to use these words a lot. Dominant, recessive, dominant, recessive. A lot of the exam is going to be involved in knowing that. So I'm going to give you some hypothetical situation. Like this thing mated with this thing, and this was the results. And then you need to be able to tell me if something was dominant or recessive based on this hypothetical situation. Of course, in my opinion, this should be easy to remember. It's all in the name, right? One of them is dominant, one of them is recessive. Any questions about the third of the fourth hypotheses? Okay. When we write these down, we use uppercase italic letters to represent dominant and lowercase italic letters to represent recessive. And honestly, just to keep things simple, you don't even need to concern yourself with the italic part, because you're probably not going to be typing this out very often. So you know what? As long as you use uppercase for dominant, lowercase for recessive, we're all good. And you will notice too a lot, like using this P as an example, if something was heterozygous, you usually see it written like this, big P, little p. And I'm almost always going to do it like that. I don't think there's any kind of rule. We just normally do it like that. So technically, if you wrote little p, big P, that would not be incorrect. So, because I've received that question before. But anyway, big P, or excuse me, big letter for dominant, lower letter for recessive. So if you haven't done the P-Lab yet, remember you need to do that. There's tables you need to fill out. So this is a good time for me to go ahead and tell you this, because it's a good time. All right, so if you have a dominant trait, that means you could be, and I'm just going to use P as an example, big P, right? You could be homozygous dominant, or you could be heterozygous, big P, little p. So for the table, you literally need to write both of those, big P, or big P, little p. Anytime you have a dominant trait, you need to write both possible genotypes on there. If you want to argue it, if you somehow know about your family history a little bit better, and you're like, oh, I'm definitely big P, let me know. Go ahead. It'd be a hard argument, but it'd be such a good mental exercise that I would definitely give you those points. If you can argue, like, no, I can tell because here's my pedigree. My grandparents had this, my parents had this, my great-grandparents had this, so obviously I must be homozygous dominant, or I must be heterozygous. Me, for example, and this is a made-up example, but let's assume I had brown eyes, and let's assume I had kids that had blue eyes. Then I can tell you for sure that I must be heterozygous. No way for my kid. Let's leave it at that, because I don't want to get into that yet. But yes. Anyway, any questions about uppercase versus lowercase, dominant versus recessive? All right. Number 4, the fourth and final hypothesis of his hypotheses that we're talking about. A sperm or egg only carries one allele for each inherited character because the two alleles for a character segregate during production of gametes. In other words, number 4 is chapter 8, right? Number 4 is what you learned when you learned about meiosis. Those homologous chromosomes get separated. You know that the chromosome number doubles in the S phase of interphase, and it gets cut in half after the first round of meiosis, then gets cut in half again. Because as you know, you only need half the genetic information if you're making sperm or egg. So there you go. Number 4, in a sense, is nothing new. Number 4 is what you learned in the last chapter. We're just bringing it up again and saying it in a different context. A sperm or egg carries only one allele. So again, for all the genes, or excuse me, cells in your body, you have 2 alleles for, I'm oversimplifying here, but 2 alleles for eye color, right? One that you got from your mom, one that you got from your dad. Now, if you were to look at your sperm or egg, whatever, if you were to look at your gametes, 50% of them would have the allele from your mom, 50% of them would have the allele from your dad. About, right? Surely some of them maybe just didn't survive, right? So you might, the number might be slightly off, but generally it should be about 50-50. Right? Any questions so far about this fourth hypothesis? Good. So finally I can get to this. This is what we call the law of segregation. So the stuff above that bullet point is the description of the law of segregation. Alleles segregate, right? Gametes only have one allele for each character. That's the law of segregation. There's definitely a study guide question on there. There might be a test question on there. But almost definitely, yeah. You need to know what the law of segregation is. So it's the alleles that are segregated. Or, as I'm going to tell you at the end of the chapter, when you think about it, those alleles are on chromosomes, right? So it's those chromosomes that are segregating. Those homologous chromosomes are separating. So another way of saying this, backing it up, yes, we're talking about alleles, but we could talk about the whole chromosome if you want, right? So again, you have, I'm going to randomly pick number 15. Why not? Of all of them, they're all the same, but for chromosome number 15, in all of your cells, you have one chromosome 15 that you got from your mom, and you got one chromosome number 15 that you got from your dad. And if you were to look at all of your sperm or eggs, about 50% of them would have only mom chromosome number 15s, and about the other 50% would have only dad chromosome number 15s, because they separated, it's segregated in meiosis. And of course, when the sperm and the egg unite in fertilization, as you learned from chapter 8, each one of them contributes its alleles, and this restores the paired condition of the offspring. So again, chapter 8 stuff. The only new thing is this bullet point right here, right? The law of segregation. So basically, I just gave a name to things you already learned from chapter 8. And when this happens, that's called the law of segregation. So any questions about this slide before I give you some visualizations to help? Okay, so this picture should look familiar. It's the same picture that I showed you earlier, except now we're just including alleles and numbers. So I almost don't like the way they did this. Oh, no, that's fine. So this thing's homozygous dominant. This thing's homozygous recessive. When this thing goes through meiosis, when both of them go through meiosis, those alleles segregate, right? One goes this way, one goes this way. It just so happens that since it's homozygous dominant, that 100%, right, all of them are going to be big P. So yes, they segregated. Your book just doesn't do a good job of showing that. But there's only now one allele, right? So we're looking at the sperm and the egg. There's only one allele. Same thing here. It's homozygous recessive. It only has little p's. So it doesn't matter which sperm, which sperm or egg you look at, they're all going to be little p because that's the only thing it has to offer. So yes, they did segregate in meiosis, but again, 100% of them are going to be little p because that's the only thing they have to offer. Therefore, without a doubt, when you cross this thing with this thing, 100% of the offspring are going to be heterozygous, right? Because they're getting half of their information from the male, half of the information from the female. So it would have to be heterozygous, 100%. All of them have to be heterozygous. Of course, when this thing goes through meiosis and those things segregate, obviously half of them are going to have the big P, half of them are going to have the little p. Does that make sense? It's the only way for it to happen, right? They segregate. So about half will have big P and then about half will have little p. And of course, I say about because, again, technically, when you go through meiosis, sometimes things go wrong, as we learned from chapter 8. So anyway, then we do this thing, which is called a Punnett square, which you have to do in the lab, right? So we look at the possibilities here. Of course, they're heterozygous, so 50% of the sperm are going to be big P, 50% of the sperm are going to be little p, 50% of the eggs are going to be big P, 50% of the eggs are going to be little p. And then you look at the possible combinations of those, right? 1/4 of the time, it's going to be big P, big P. 1/4 of the time, it'll be big P, little p. Another 1/4 of the time, it'll be big P, little p, right? You bring those together. And then 1/4 of the time, it'll be little p, little p. That's a Punnett square. And of course, these particular numbers, the ones I told you need to know, that ratio, 3-to-1 phenotypic ratio for the F2 generation, and a 1-to-2-to-1 genotype ratio, because it just so happens this F2 generation happened to be heterozygous. I'll need to bring that up because for what we're talking about, that's fine. Just leave it at that. But in the context of the lab we're doing, remember, sometimes you're not going to be mixing a heterozygous and a heterozygous. You might be mixing a heterozygous with a homozygous dominant, or a heterozygous with a homozygous recessive, or a homozygous dominant with a homozygous recessive, or homozygous dominant with a homozygous dominant. So we'll come back to all that a little bit later, but there you go. Any questions about this slide? Again, you need to recognize this 3-to-1 ratio, phenotypic ratio, and a 1-to-2-to-1 genotype ratio. as the results of the monohybrid cross. Punnett square. I just showed you. Now I can see it in writing. A Punnett square highlights the 4 possible combination of gametes, which then would obviously lead to the 4 possible type of offspring that you would get. And each square represents equally the probable product of fertilization. So that's all that is. I'm just putting in writing what I just showed you right here, which is what a Punnett square is. The next word for attendance will be, actually, I won't even say it. I'll just circle it. So there you go. Any questions? Okay, let's talk about genetic alleles and homologous chromosomes. I've already been talking about this as I was bringing these terms up. I was already reminding you of what you've already learned in Chapter 8, but let's officially put this on paper, so to speak. Some other terms you definitely need to know, especially for lab, is phenotype and genotype. Phenotype is the physical appearance. So when we're talking about characters and traits, that's what we're talking about, right? As a purple flower or a white flower, that's the phenotype. Or in lab, you have a smooth chin or a dimple chin. That's your phenotype. Of course, there's so many different ways to describe the phenotype as well. Go ahead and say that too. So when you're doing the lab and you're putting in your phenotype, there's so many ways you can describe it. So again, for the chin, you could say smooth chin or dimple chin. Or you could say dominant phenotype or recessive phenotype. Whatever. All describing the same thing. Genotype, on the other hand, it's a little bit more specific. The genotype is the genetic makeup that results in the phenotype. So it's what genes, it's which alleles you have that results in the phenotype. And again, like I've already said, the 3 different genotypes are homozygous dominant, which is when you have 2 dominant alleles, heterozygous, which is when you have a dominant allele and a recessive allele, and homozygous recessive, which is when you have 2 recessive alleles. And again, your genotype determines your phenotype. This suffices for now, but later I will bring this up. It's not always physical appearance. Just to keep things simple, we talk about physical appearance, but sometimes your phenotype is like a disease. You either have the disease or you don't. You might not necessarily see anything, but, or like, the increased risk for breast cancer, for example. You can't see that in somebody, but that is a phenotype. Anyway, each character in his studies have the same inheritance patterns, which is that the parental trait disappeared in the F1, right? So if we're talking about the recessive, we had a parental trait disappear in the F1. So again, using the flowers as an example, the white trait disappeared. And then it reappeared again in that next generation, about 25% of them, right? So again, using the flowers as an example, about 1/4 or about 25% of the offspring had that white trait, that white flower again. So another way of saying this is it's that 3-to-1 ratio, right? 3 dominant, 1 recessive. Or you could even say 25%, 75% to 25%, right? It's all describing the same situation in different ways. Anyway, any questions about this slide? All right. The next slide I'm going to show you shows a pair of homologous chromosomes, which you learned all about in the last chapter. But to remind you, they are chromosomes that have alleles of the same genes. And then a new term, even though I've been describing this before, I've not given you this term, which is called gene locus. That is the specific location of a gene on a chromosome. So earlier today, I used some hypothetical, incorrect example of the bad breath gene. I said if it was on the middle of chromosome number 5, right? I said if it was on the middle of chromosome number 5 for me, it would be for him and her and all of you, right? Because they all genes have the same gene locus on the same chromosome. At least the way I teach it, at least the way we're teaching it. There are exceptions to that. You can look up for independent work, but to keep things simple, that will suffice. Just know that there are exceptions to everything I'm teaching. So the alleles for a gene reside, like I said, same location on the homologous chromosomes. And of course, obviously, they may be identical alleles or different ones. So you might have 2 alleles for stinky breath, or you might have an allele for stinky breath and an allele for good smelling breath, or you might have 2 alleles for good smelling breath. And here's a picture of what we're talking about. We're just looking at one particular chromosome. We got one version from your mom, one from your dad. We're just happen to look at 3 different genes, gene P, gene A, and gene B on this chromosome. In this case, we can see that they're homozygous dominant because both of the alleles are the same. In this case, they're homozygous recessive. Again, both of the alleles are the same. In the case of gene B, you got a dominant there and a recessive there. And I guess the last word for attendance, even though we just did one, won't even do a word. Let's just do a letter. B. B is the last letter for attendance. Anybody have any questions about anything? All right. If I didn't see you on Monday in lab, I hope to see you today.